You are listening to this right now, and something extraordinary is happening. The words arriving through your ears are being converted into electrical signals by tiny hair cells in your cochlea, traveling along the auditory nerve at speeds of up to 70 meters per second, arriving at the brainstem, being relayed to the thalamus, being projected to the auditory cortex in your temporal lobe, being parsed for meaning in Wernicke's area, being connected to memories and associations in networks spread across your entire brain, and producing the experience of understanding. And somewhere in the process, a part of your brain is watching all of this happen, aware that it is happening, curious about what comes next. That is the brain. It is the only object in the known universe that can examine itself. It is 3 pounds of soft, pale tissue, the consistency of firm tofu, drawing about 20% of the body's energy on just 2% of its mass, running at the same power as a 20-watt light bulb, and producing everything you have ever thought, felt, seen, remembered, imagined, and understood. It contains approximately 86 billion neurons, and those neurons are connected to each other by approximately 100 trillion synapses, junctions where one neuron communicates with another. If you were to lay all the neural wiring in a single human brain end to end, it would stretch approximately 100,000 kilometers, nearly 1/4 of the distance from the Earth to the Moon. Tonight, as you rest, your brain will not stop. It will slow down. It will change its pattern of activity. It will do things during sleep that it cannot do while you are awake. Things that are essential for your memory, your health, and your ability to think clearly tomorrow. And the better you understand what your brain is doing right now, as you settle into stillness, the more you can appreciate the quiet, extraordinary process that is already beginning. Let your breathing slow. The brain responds to slow breathing. It is not a metaphor. The vagus nerve carries signals from the body to the brainstem, and slow, deep breathing activates parasympathetic pathways that reduce cortisol, lower heart rate, and shift the brain away from the high-frequency beta waves of active thought toward the slower alpha waves of relaxed awareness. Every slow breath you take right now is a signal to your brain that it is safe to begin the transition toward sleep. The brain is listening. The neuron is the fundamental unit of the nervous system, and it is one of the most remarkable cells in the biology of life. A typical neuron has a cell body called the soma, which contains the nucleus and manages the cell's metabolic functions. Extending from the soma are dendrites, branching tree-like extensions that receive incoming signals from other neurons, sometimes thousands of them simultaneously. A single neuron in the cerebral cortex may have dendrites that receive signals from up to 200,000 other neurons. Extending from the soma in the other direction is the axon, a single long process that can range in length from less than a millimeter in local interneurons to more than a meter in the motor neurons that extend from the spinal cord to the muscles of the foot. The axon is often wrapped in a white, fatty insulating sheath called myelin, produced by specialized cells called oligodendrocytes in the brain and Schwann cells in the peripheral nervous system. The myelin sheath is interrupted at regular intervals by small gaps called nodes of Ranvier, and it is at these nodes that the electrical signal jumps from node to node in a process called saltatory conduction, dramatically increasing the speed at which signals travel. The action potential is the fundamental electrical event of the nervous system. At rest, a neuron maintains a voltage difference across its membrane of about -70 millivolts, the inside of the cell being more negative than the outside, maintained by ion pumps that continuously pump sodium ions out and potassium ions in. When the dendrites receive enough excitatory signals from other neurons to depolarize the membrane at the axon hillock, the zone where the axon meets the soma, to a threshold of about -55 millivolts, a rapid cascade occurs. Voltage-gated sodium channels snap open, sodium ions rush into the cell down their concentration gradient, and the membrane potential shoots from -55 to +40 millivolts in less than a millisecond. This depolarization then triggers the opening of sodium channels in the adjacent patch of membrane, and the signal propagates down the axon as a wave of depolarization. In myelinated axons, this process jumps from node to node and travels at speeds of up to 120 meters per second. In the thinnest unmyelinated fibers, signals travel as slowly as 0.5 meters per second. The brain uses this enormous range of conduction velocities to integrate information arriving from different distances at approximately the same time. At the end of the axon, the signal arrives at the synaptic terminal, the terminal boton, where the neuron communicates with the next cell. The synaptic terminal contains thousands of small membrane-bound sacs called synaptic vesicles, each containing a specific neurotransmitter molecule. When the action potential arrives, voltage-gated calcium channels open, calcium floods into the terminal, and the vesicles fuse with the terminal membrane and release their neurotransmitter contents into the synaptic cleft. A gap of only about 20 nanometers between the sending and receiving cells. The neurotransmitter molecules diffuse across this gap in less than a millisecond and bind to specific receptor proteins on the membrane of the receiving cell, causing changes in the receiving cell's membrane potential that either make it more likely to fire an excitatory postsynaptic potential or less likely to fire an inhibitory postsynaptic potential. The receiving cell integrates thousands of such inputs simultaneously, and if the sum of excitatory inputs exceeds the inhibitory inputs by enough to bring the membrane to threshold, it fires its own action potential. The chemistry of the brain is written in neurotransmitters. Glutamate is the most common excitatory neurotransmitter in the brain, used by the vast majority of neurons that excite other neurons. GABA, gamma-aminobutyric acid, is the most common inhibitory neurotransmitter, the brain's primary brake system. The balance between glutamate and GABA activity determines the overall excitability of neural circuits, and many drugs, from benzodiazepines to alcohol, exert their calming effects by enhancing GABA activity or reducing glutamate activity. Dopamine is a neurotransmitter associated with reward, motivation, and learning, released by neurons in the ventral tegmental area and substantia nigra, and projecting to the striatum and prefrontal cortex. It is not, as is sometimes said, simply a pleasure chemical. Its primary function is to signal the prediction and receipt of reward, teaching the brain which actions lead to good outcomes. Serotonin is involved in mood regulation, sleep, appetite, and social behavior, and most of the brain's serotonin is produced by a small cluster of neurons in the brainstem called the Raphe nuclei. Norepinephrine regulates arousal and alertness, and its activity drops precipitously during sleep, which is one reason why the sleeping brain processes information differently from the waking brain. Acetylcholine is involved in memory, attention, and muscle contraction, and its release during REM sleep is thought to be involved in the vivid dreaming that occurs in that stage. Here is where the brain becomes something stranger than any machine we have built. The four major lobes of the cerebral cortex, the deeply folded outer layer of the brain that is responsible for most of what we consider distinctly human cognition, each have specialized functions that were mapped over more than a century of careful observation of brain injuries, surgical experiments, and more recently, functional brain imaging. The frontal lobe, occupying the forward third of the brain behind the forehead, is the seat of personality, decision-making, planning, impulse control, and the regulation of emotions. The case of Phineas Gage, a railroad construction foreman in Vermont, who in 1848 survived the passage of a tamping iron 3 centimeters in diameter and more than a meter long through his left frontal lobe, is perhaps the most famous in all of neuroscience. Before the accident, Gage was described by those who knew him as a reliable, responsible, and well-liked worker. After the accident, while his language, memory, and motor function were largely intact, his personality was transformed. He became fitful, irreverent, profane, and unable to carry out plans he had made, abandoning them as soon as something else caught his attention. The damage to his frontal lobe had disconnected the cognitive machinery of planning and decision-making from the inhibitory and social functions that normally constrain behavior. Gage's case established, for the first time in neurological history, that personality and social behavior are products of specific brain structures, not of some non-physical soul. The parietal lobe, behind the frontal lobe and above the occipital lobe, processes spatial information, integrates sensory input from different modalities, and contains the primary somatosensory cortex, the strip of cortex that represents the body surface. The somatosensory cortex is organized in a body map called the homunculus, in which different body parts are represented in proportion to their sensory importance rather than their physical size. The hands and lips occupy a much larger area than the torso or legs, because the density of sensory receptors in the hands and lips is far higher. The temporal lobe on the sides of the brain beneath the parietal lobe is involved in auditory processing, language comprehension, and memory. It contains the hippocampus, one of the most studied and most important structures in the brain, buried in the medial temporal lobe like a small seahorse, which is what its name means in Greek. The occipital lobe at the back of the brain is devoted almost entirely to visual processing. It contains the primary visual cortex, which receives input from the eyes via the thalamus, and the surrounding visual association areas, which process color, motion, depth, object recognition, and face recognition. The cerebellum, the small, densely folded structure at the back and base of the brain, contains roughly 69 billion neurons, more than 80% of all the neurons in the entire nervous system, despite accounting for only about 10% of the brain's volume. The cerebellum coordinates movement, balance, and fine motor control, and is involved in procedural memory, the kind of memory that stores skills like riding a bicycle or playing a piano scale, The cerebellum does not initiate movements. It receives copies of movement commands from the motor cortex and compares them to sensory feedback from the muscles and joints, generating error correction signals that smooth and coordinate the movement. When the cerebellum is damaged, movements become ataxic, clumsy, uncoordinated, and inaccurate, not paralyzed, but imprecise. A person with cerebellar damage can reach for a glass, but their hand will overshoot and undershoot in oscillating corrections, unable to stop smoothly at the target. And here is something that will change how you think about your own memory. In 1953, a 27-year-old man named Henry Molaison, known in the scientific literature as HM, until his death in 2008, underwent surgery to treat his severe epilepsy. The surgeon, William Beecher Scoville, removed a substantial portion of Henry's medial temporal lobes on both sides of the brain, including most of the hippocampus. The surgery was successful at reducing his seizures, but when Henry woke up, something had changed in a way no one had anticipated. He could no longer form new long-term memories. He could remember events from before the surgery, though his memory for the years immediately preceding the surgery was impaired. And he could hold information in his conscious awareness for a minute or two, but the moment his attention was diverted, the new information was gone completely. He could meet a doctor, have a long conversation, and then 2 minutes later have no memory that the meeting had ever occurred. He could read the same magazine article hundreds of times without any sense of having seen it before. He lived in a perpetual present, unable to accumulate the experiences that normally constitute a life. Henry Molaison was studied continuously for 55 years after his surgery, and he became the most studied individual in the history of neuroscience. Among the many discoveries made through studying him was the distinction between declarative memory, the memory for facts and events that requires the hippocampus, and procedural memory, the memory for skills and habits that is stored in the cerebellum and basal ganglia and does not require the hippocampus. Henry could learn new motor skills. Over repeated sessions, he improved at a mirror tracing task, drawing a figure while watching his hand in a mirror, getting better each time, even though he had no memory of having practiced before. His procedural memory system was intact. Only his ability to form new declarative memories was destroyed. This dissociation showed that memory is not a single thing stored in a single place, but a collection of distinct systems that operate differently and are stored in different structures. The hippocampus forms memories by changing the strength of the synaptic connections between neurons in a process called long-term potentiation. Discovered by Timothy Bliss and Terry Elmo in 1973, long-term potentiation is the persistent strengthening of a synapse following repeated stimulation. When a synapse is stimulated repeatedly, the receiving cell begins to produce more AMPA receptors in its membrane at that synapse, making it more sensitive to future stimulation from the same sending cell. The key molecular player in this process is the EMDA receptor, a glutamate receptor that acts as a molecular coincidence detector. It only opens when glutamate is bound to it and the receiving cell's membrane is already depolarized by other inputs. This coincidence detection is thought to be the cellular mechanism of Hebb's rule. The observation made by the Canadian psychologist Donald Hebb in 1949 that neurons that fire together wire together, synapses between cells that are simultaneously active are strengthened, while synapses between cells that are not simultaneously active are weakened or eliminated. The brain you are resting in right now is not the same brain it was a year ago. It is not even quite the same brain it was this morning. Every experience you have, every thought, every emotion, every skill you practice changes the physical structure of the synaptic connections in your brain. This is neuroplasticity, the brain's ability to reorganize itself by forming new neural connections throughout life. Neuroplasticity is not unlimited. Some connections are more easily formed at certain ages than others, and some brain functions become less plastic with age. But the adult brain retains a remarkable capacity for change, and this capacity is exercised every day, including during sleep, when the brain consolidates the experiences of the day into more stable memory traces. The default mode network is one of the most important discoveries of modern neuroscience. Before its discovery in 2001 by Marcus Rachel and his colleagues at Washington University in St. Louis, the brain's activity during rest was considered simply the absence of the activity observed during tasks. Rachel noticed that when subjects were asked to perform a demanding cognitive task, specific regions of the brain decreased their activity relative to rest, and these regions were always the same: the medial prefrontal cortex, the posterior cingulate cortex, the angular gyrus, and parts of the temporal lobe. He concluded that these regions were not simply quiet during task performance, but were specifically and consistently active during rest, forming a network he called the default mode network. The default mode network is now understood to be involved in mind wandering, self-referential thought, mental simulation of future and past events, and social cognition, imagining other people's mental states. When you are not focused on an external task, when you are lying quietly and letting your mind drift, the default mode network is active, running its internal processes, connecting experiences, constructing and updating the narrative of the self. The brain's sleeping activity is organized into waves. Brain waves are rhythmic electrical oscillations produced by the synchronized activity of large populations of neurons, measurable from electrodes placed on the scalp in the technique called electroencephalography, or EEG. Different brain states produce different characteristic frequencies of oscillation. When you are alert and actively thinking, the predominant waves are beta waves, with frequencies between about 13 and 30 cycles per second, and gamma waves at 30 to 100 cycles per second. When you close your eyes and relax but remain awake, alpha waves appear at 8 to 12 cycles per second, producing a characteristic sense of calm, unfocused awareness. As you begin to fall asleep, the brain transitions through theta waves at 4 to 8 cycles per second, and finally into the delta waves of deep sleep at 0.5 to 4 cycles per second, the slowest and most synchronized oscillations the brain produces. Sleep itself is organized into cycles of roughly 90 minutes, each containing several distinct stages. The first stage, NREM 1, is the lightest sleep, easily interrupted, characterized by the slowing of brain waves into theta frequencies. This is the stage in which hypnic jerks occur, those sudden muscle contractions that sometimes wake you just as you are drifting off, believed to be caused by the brain misinterpreting the relaxation of falling asleep as the beginning of a fall and sending a sudden motor correction signal. The second stage, in NREM 2, is characterized by the appearance of sleep spindles, bursts of high-frequency oscillations at 12 to 14 cycles per second, lasting about a second each, and K complexes, large, slow waves that punctuate the EEG. Sleep spindles are generated by circuits in the thalamus and are thought to be involved in the consolidation of declarative memories, the memories for facts and events that the hippocampus processes. The third stage, NREM 3, is deep sleep, also called slow wave sleep, characterized by the large amplitude delta waves that represent the most synchronized activity the brain produces. It is hardest to wake from and most important for physical restoration, immune function, and the consolidation of certain types of memory. After the first cycle of NREM sleep, the brain transitions briefly back toward wakefulness and then into REM sleep, rapid eye movement sleep, in which the brain's electrical activity resembles wakefulness, but the body is paralyzed, a state called atonia, produced by the active inhibition of the spinal motor neurons by neurons in the brainstem. This paralysis prevents the dreaming brain from acting out its dreams. People with REM behavior disorder, in which the atonia mechanism is disrupted, do act out their dreams, and injuries result. REM sleep is when the most vivid dreaming occurs, and it appears to be important for emotional memory processing, creativity, and the integration of new information with existing knowledge structures. The ratio of NREM to REM sleep changes across the night. The early cycles of the night contain more NREM 3 deep sleep, and the later cycles contain more REM sleep, which is why sleep deprivation that cuts the last few hours disproportionately reduces REM sleep. Memory consolidation during sleep is one of the most actively studied areas of neuroscience. The general theory, well supported by experimental evidence, is that during waking life, the hippocampus rapidly encodes new experiences in its highly plastic synaptic connections, and during sleep, particularly during NREM sleep, these hippocampal memory traces are transferred to the neocortex, where they are integrated with existing knowledge structures and stored in a more stable, distributed form. The mechanism involves sharp wave ripples in the hippocampus, brief high-frequency bursts of activity during which the hippocampus replays the day's experiences in compressed form, and the coupling of these ripples to sleep spindles generated by the thalamus and to slow oscillations in the neocortex, and a precise temporal coordination that drives the transfer of information. Experiments in which specific memories were reactivated during sleep by presenting subjects with odors associated with a learning task during deep NREM sleep produced enhanced memory for the associated task the next morning, demonstrating that this replay and consolidation process is not simply random, but can be targeted. And here is where the story of sleep and the brain takes a turn that no one anticipated before 2013. The brain, uniquely among the organs of the body, lacks a traditional lymphatic system, the network of vessels that in other tissues drain cellular waste products away from the interstitium and delivers them to the liver for processing and disposal. For decades, this was considered a puzzle. The brain is the organ with the highest metabolic rate, producing the most waste products per unit of tissue, yet it appeared to lack any mechanism to remove those waste products. In 2013, the neuroscientist Micah Nedergaard and her colleagues at the University of Rochester published a discovery that resolved this puzzle and transformed the neuroscience of sleep. They described a system they called the glymphatic system, a network of channels surrounding the blood vessels of the brain through which cerebrospinal fluid flows during sleep, flushing out the metabolic waste that accumulates in the brain's interstitial space during waking activity. During sleep, the interstitial space of the brain expands by about 60% compared to waking, allowing cerebrospinal fluid to flow much more easily through it. The glymphatic system clears, among other substances, amyloid beta, the protein fragment that accumulates in the brains of Alzheimer's patients, and tau, another protein whose abnormal accumulation is associated with neurodegenerative disease. One night of sleep deprivation produces a measurable increase in amyloid beta in the human brain, and chronic sleep deprivation is associated with significantly elevated amyloid beta burden. Sleep is not passive recovery. It is active neural maintenance. Right now, as you listen and your body stills, that process is beginning. The glymphatic system is starting to work. The slow waves of sleep, when they come, will drive the pulsing flow of cerebrospinal fluid through the channels around the brain's blood vessels and the waste products of a day's worth of metabolic activity. The molecular debris of everything your brain has done today will be carried away. The consciousness that is listening to these words is one of the deepest puzzles in all of science. The hard problem of consciousness, named by the philosopher David Chalmers in 1995, is the question of why and how physical processes in the brain give rise to subjective experience at all. It is relatively easy to explain, at least in principle. The functional aspects of consciousness, why the brain processes information in the way it does, why it integrates sensory input, maintains working memory, generates behavior. But why is there something it is like to be a brain? Why do the electrochemical processes in 86 billion neurons produce the experience of reading these words, the feeling of the air on your skin, the sense of a self doing the reading? No one knows the answer. The global workspace theory, proposed by Bernard Bears and developed by Stanislas Dehaene and Jean-Pierre Changeux, suggests that consciousness arises when information is broadcast widely across the brain through a global workspace, a network of neurons in the frontal and parietal cortices that can receive and rebroadcast information from specialized processing areas. On this view, you are conscious of something when its representation is amplified and broadcast to the whole brain, allowing it to influence many different cognitive processes simultaneously. The integrated information theory, proposed by the neuroscientist Giulio Tononi, takes a different approach. It proposes that consciousness is identical to integrated information, measured by a quantity called phi, and that any system that integrates information in a specific way has some degree of consciousness, with the amount proportional to the phi of the system. Both theories have empirical predictions and active research programs, and neither is yet established as correct. The split brain experiments of Roger Sperry and Michael Gazzaniga in the 1960s revealed that the two hemispheres of the brain can operate as semi-independent systems when the corpus callosum, the massive bundle of about 200 to 250 million nerve fibers that connects the left and right hemispheres, is severed. This surgery, callosotomy, was performed in the 1960s to treat severe epilepsy in patients for whom other treatments had failed. After the surgery, patients appeared normal in everyday life. They could walk, talk, and behave normally, but under controlled experimental conditions, Sperry and Gazzaniga showed that the two halves of the brain could hold different information, make different decisions, and even have different beliefs, without either half being aware of what the other half knew. Information presented only to the left visual field, processed by the right hemisphere, could not be named by the patient because language is in the left hemisphere, but the patient could pick out the corresponding object with the left hand, controlled by the right hemisphere. The right hemisphere knew the answer, it just could not say it. These experiments showed that unified conscious experience is not a fixed property of a brain, but something constructed by the brain, something that can be disrupted by damage, and something that can, in an important sense, be split. The mirror neuron system is one of the most discussed and most misrepresented discoveries in modern neuroscience. Mirror neurons were first discovered by Giacomo Rizzolatti and his colleagues at the University of Parma in 1992, while recording from individual neurons in area F5 of the macaque-monkey promoter cortex. They found neurons that fired both when the monkey performed a specific action, such as grasping a piece of food, and when the monkey observed another individual performing the same action. The neurons mirrored the observed action in the monkey's own motor system. In humans, evidence from neuroimaging suggests a mirror system in Broca's area in the frontal lobe and in the inferior parietal lobule. The mirror neuron system has been proposed as a neural substrate for imitation, understanding the actions and intentions of others, and empathy. Some have proposed it as a key mechanism in autism spectrum disorder, suggesting that a deficient mirror system might underlie the social and imitative difficulties associated with autism, a hypothesis called the broken mirror theory. The evidence for this specific hypothesis is disputed, but the broader role of mirror, like computations in social cognition, remains an active area of research. The brain contains approximately 100 billion blood vessels, including capillaries so narrow that red blood cells must squeeze through single file. These capillaries are lined by endothelial cells that are connected by extremely tight junctions, forming the blood-brain barrier, a selective filter that protects the brain from pathogens, toxins, and the fluctuations in blood composition that would interfere with the precise ionic environment that neurons require. The blood-brain barrier excludes more than 98% of all molecules, including most drugs, which is why treating brain diseases with drugs is so challenging. The drugs must either be small and lipid-soluble enough to pass through the barrier, be actively transported across it by specific transport proteins, or be administered in ways that bypass it. The brain makes exceptions at specific locations called circumventricular organs, small regions where the blood-brain barrier is deliberately absent, allowing the brain to sample blood composition directly and regulate functions like thirst, blood pressure, and hormone release. The teenage brain is a brain in transition. The myelination of axons, The process of wrapping axons in myelin to speed their conduction proceeds in a roughly posterior to anterior direction during development. The primary sensory and motor cortices are fully myelinated in childhood, while the prefrontal cortex, the last region to complete myelination, does not reach full adult development until approximately age 25. During adolescence, the prefrontal cortex is the least mature region of the brain, and its connections to the limbic system, which generates emotional and reward-related signals, are still being refined. The result is a brain with full emotional reactivity and reward sensitivity, but incomplete executive control, a powerful engine without a fully developed braking system. This is not a defect, but a developmental stage shaped by evolution that produces the risk-taking, novelty-seeking, and social sensitivity that characterize adolescence and that may have been adaptive in ancestral environments where young individuals needed to venture into new territories and form new social bonds. The brain you are resting in tonight is a connectome, a map of all the connections between all its neurons that is unique in all the universe. No two human brains are wired in exactly the same way. Development, experience, and genetics combine to produce a configuration of 86 billion neurons and 100 trillion synapses that has never existed before and will never exist again. Sebastian Sung, a physicist turned neuroscientist, has estimated that the full human connectome, if mapped at the level of individual synapses, would contain approximately 2.5 petabytes of information, roughly equivalent to 2.5 million gigabytes. A project to map 1 cubic millimeter of mouse cerebral cortex produced 100 terabytes of data from a tissue volume the size of a grain of sand. The full human connectome remains a distant goal, but its mapping, when it is eventually achieved, will be one of the great scientific accomplishments of human civilization. The brainstem, the stalk of neural tissue connecting the brain to the spinal cord, is the most ancient part of the brain in evolutionary terms, and it performs functions so fundamental that damage to it is rapidly fatal. The medulla oblongata, the lowest part of the brainstem, contains the neural circuits that control breathing, heart rate, and blood pressure. Rhythmic breathing is generated by a group of neurons in the medulla called the pre-Botzinger complex, which fires in a rhythmic pattern that drives the respiratory muscles even when the rest of the brain is entirely inactive. The pons, above the medulla, contain circuits involved in sleep regulation, including the generation of REM sleep and the atonia that accompanies it. The midbrain contains the substantia nigra and the ventral tegmental area, the major dopamine-producing nuclei, which project to the striatum and prefrontal cortex and are essential for motivation, reward learning, and movement. Damage to the substantia nigra's dopamine neurons causes Parkinson's disease, with its characteristic tremor, rigidity, and bradykinesia. The thalamus, a paired egg-shaped structure at the center of the brain, is often described as the brain's relay station. Nearly all sensory information, except olfactory signals, passes through the thalamus before reaching the cortex. But the thalamus is far more than a simple relay. It actively gates the flow of information to the cortex, amplifying some signals and suppressing others based on the brain's current state of alertness and attention. During sleep, The thalamus generates sleep spindles and plays a critical role in preventing sensory signals from waking the cortex. It is the thalamus that allows you to sleep through familiar background noises while still waking you to your own name or to a baby's cry. The thalamus and cortex are connected by reciprocal loops, and the oscillations generated by these thalamocortical circuits are thought to be a fundamental mechanism of consciousness, with the loss of thalamocortical connectivity being associated with loss of consciousness in anesthesia and coma. The basal ganglia, a collection of nuclei deep in the brain, including the caudate nucleus, putamen, globus pallidus, and subthalamic nucleus, are involved in the selection and initiation of actions, habit formation, and the learning of procedural skills. The basal ganglia receive input from all areas of the cortex and project back to the cortex through the thalamus, forming loops that are thought to select which actions to execute by inhibiting competing actions and releasing the selected action from inhibition. When habits are formed through repeated practice, the neural representations of the habit gradually transfer from the hippocampus, which initially stores all new learning, to the basal ganglia, where they are stored as automatic, rapid routines that can be executed without conscious attention. This is why well-practiced skills like typing, driving on familiar roads, or playing a musical passage you have practiced thousands of times can be performed while conscious attention is directed elsewhere. The hypothalamus, a small but extraordinarily powerful structure just below the thalamus, controls the brain's homeostatic functions, body temperature, hunger, thirst, circadian rhythms, sleep-wake cycles, and the release of hormones from the pituitary gland. It contains the suprachiasmatic nucleus, a pair of tiny clusters of neurons just above the optic chiasm, where the optic nerves from the two eyes cross. The suprachiasmatic nucleus is the brain's master clock, maintaining a circadian rhythm of approximately 24 hours in the absence of any external time cues through the self-sustaining oscillation of a set of clock genes. The products of these genes inhibit their own transcription with a delay of several hours, producing a molecular oscillation that drives the rhythmic activity of suprachiasmatic neurons and, through their connections to other brain regions and to the pineal gland, coordinates the timing of virtually every physiological process in the body. The pineal gland, a small pine cone-shaped structure in the center of the brain that Descartes famously identified as the seat of the soul, receives signals from the suprachiasmatic nucleus about the light-dark cycle and secretes melatonin in the dark. Melatonin is not a sleep-inducing drug. It is a timing signal that tells the body what time of day it is. Light exposure suppresses melatonin secretion, which is why exposure to blue wavelength light from screens in the hour before bed delays the onset of sleep. The brain interprets the blue light as daylight and delays the melatonin signal. The cells in the retina that project to the suprachiasmatic nucleus are not the rods and cones used for vision, but a third type of photoreceptor. Intrinsically photosensitive retinal ganglion cells, which express a photopigment called melanopsin that is most sensitive to blue wavelengths around 480 nanometers. These cells are exquisitely sensitive to the blue wavelength component of ambient light, and the signal they send to the suprachiasmatic nucleus is the primary and training input that keeps the circadian clock synchronized to the light-dark cycle. Synesthesia is a condition in which stimulation of one sensory modality automatically and involuntarily triggers a perception in another sensory modality. The most common form, grapheme color synesthesia, is the consistent perception of specific colors when viewing letters or numbers. The letter A might always appear red, the number 7 always blue. Other forms include chromesthesia, in which sounds trigger color perceptions, lexical gustatory synesthesia, in which words trigger specific taste sensations, and mirror touch synesthesia, in which observing another person being touched triggers the sensation of being touched in the same location. Synesthesia occurs in roughly 4% of the population to some degree, and it runs in families. The neurological explanation involves enhanced connectivity between adjacent cortical areas. In grapheme color synesthesia, the color area V4 is abnormally connected to or activated by the letter processing area in the fusiform gyrus, which lies adjacent to it in the temporal lobe. Vladimir Nabokov, the novelist, was a grapheme color synesthete who described the individual colors of each letter of the alphabet in his autobiography Speak Memory, and reported that his wife and son were also synesthetes. The phantom limb phenomenon, the vivid and often painful sensation of a limb that has been amputated, is one of the most compelling demonstrations of the brain's constructive role in perception. The somatosensory cortex maintains a map of the body, the homunculus, and when a limb is amputated, the region of the map corresponding to that limb no longer receives input, but the neurons in that region do not simply go silent. They are taken over by neighboring regions of the map in a process of cortical reorganization that can occur within weeks of amputation. The hand area of the somatosensory cortex is adjacent to the face area, and in patients with arm amputation, touching different parts of the face can trigger sensations referred to the missing hand, with different spots on the face corresponding to specific fingers of the phantom hand in a stable and consistent mapping. The neuroscientist V.S. Ramachandran used this insight to develop the mirror box, a simple device in which a mirror placed vertically between the patient's two arms allows the patient to see the reflection of the intact arm in the position of the missing arm. When the patient moves the intact arm, the visual feedback of seeing the reflection, apparently a moving hand in the phantom's position, can relieve phantom limb pain by providing the missing arm area of the motor cortex with the visual confirmation that the movement command has been executed. The binding problem is the question of how the brain combines the outputs of dozens of separate processing areas into a single, unified perceptual experience. When you look at a red ball rolling across the floor, your visual cortex processes the color in one region, the motion in another, the shape in another, and the depth in another. Yet you experience not separate qualities, but a single unified percept, a red, round, moving ball. How the brain binds these separately processed features into a unified experience is unknown. The most influential hypothesis is that binding is achieved by the synchronized oscillation of neurons representing different features at gamma frequencies. Around 40 cycles per second, Neurons in different brain areas that fire together in synchrony are bound into a common perceptual object. This gamma synchrony hypothesis has experimental support from recordings in animals and from EEG studies in humans, but it also has critics who argue that gamma synchrony is not sufficient to explain the unity of conscious experience. The neuroscience of pain involves both the detection of tissue damage by peripheral nociceptors, specialized sensory neurons that respond to noxious stimuli, and the modulation of pain signals by descending pathways from the brain that can dramatically increase or decrease the intensity of pain experienced. The periaqueductal gray, a region of the midbrain, is the primary source of descending pain modulation. It receives inputs from the prefrontal cortex and limbic system and sends signals to the dorsal horn of the spinal cord that can inhibit the transmission of pain signals before they reach the brain. The endogenous opioid system, consisting of endorphins, enkephalins, and dynorphins that bind to the same receptors as morphine and other opioid drugs, is the primary molecular mechanism of this descending pain control. Intense exercise, sustained focus, meditation, and the placebo effect all activate the endogenous opioid system and produced analgesia, reduced sensitivity to pain. The expectation of pain relief is itself enough to trigger the release of endorphins, which is the mechanism of the placebo response. The gut contains approximately 100 million neurons, forming the enteric nervous system, sometimes called the second brain. The enteric nervous system can function independently of the central nervous system. It controls the complex, coordinated movements of digestion and the secretion of digestive enzymes without input from the brain or spinal cord. But it is also in constant two-way communication with the brain via the vagus nerve, which carries signals both from the brain to the gut and, crucially, from the gut to the brain. Approximately 80 to 90% of the fibers in the vagus nerve carry signals upward from the gut to the brain, not downward. The gut-brain axis, the communication between the gut microbiome, the enteric nervous system, and the central nervous system, influences mood, anxiety, and cognitive function in ways that are only beginning to be understood. The gut produces about 95% of the body's serotonin in enteroendocrine cells in the gut lining, where it regulates intestinal movement rather than mood directly. But the gut microbiome influences the production of neurotransmitter precursors and short-chain fatty acids that cross the blood-brain barrier and affect brain function. Adult neurogenesis, the generation of new neurons in the adult brain, was once thought to be impossible. The dogma of 20th century neuroscience held that you were born with all the neurons you would ever have, and that the loss of neurons through aging or injury was irreversible. This dogma was overturned in 1998 when Peter Erickson and Fred Gage demonstrated using a chemical marker called BIRD-U that new neurons are generated in the hippocampus of adult humans throughout life. The dentate gyrus of the hippocampus is one of only 2 regions of the adult mammalian brain in which neurogenesis reliably occurs, the other being the olfactory bulb. The rate of neurogenesis in the dentate gyrus is estimated at approximately 700 new neurons per day in the adult human hippocampus, and this rate is sensitive to environmental factors. Exercise is the most potent known stimulator of adult hippocampal neurogenesis, increasing the rate by several fold in animal studies, while stress, sleep deprivation, and alcohol suppress it. The new neurons generated in the dentate gyrus are thought to contribute to the hippocampus's ability to form new memories and to distinguish between similar experiences, a function called pattern separation. The neuroscience of music illustrates how deeply the brain's responses to sound are integrated with emotion, memory, and motor systems. Music activates a remarkably broad network of brain regions. The auditory cortex processes the acoustic properties of sound. The motor cortex and cerebellum respond to rhythm even without overt movement. The limbic system generates emotional responses, including the chills or frisson that some people experience at a particularly moving musical passage, and the memory systems store and retrieve the vast library of musical experiences accumulated over a lifetime. The release of dopamine in the nucleus accumbens, the brain's primary reward center, has been measured during music listening using positron emission tomography and peaks at the moment of maximal emotional response to music. The brain's motor system is entrained by regular rhythmic beats, which is why music with a clear beat is difficult not to move to. The beat activates motor planning circuits that produce an urge to synchronize with the rhythm. The slow, regular rhythms of ambient music, ocean waves, rain, or a gentle voice reading at a measured pace all tend to reduce brain arousal because the brain has circuits that match the rate of environmental rhythms. The thalamocortical oscillations that generate brain waves are influenced by external rhythmic stimulation in a process called entrainment, a steady rhythm at delta frequencies 0. 5 to 4 cycles per second can nudge the brain's own oscillations towards slower, more synchronized states. This is partly why a slow, calm voice is soothing, and partly why listening to slow music or rain sounds facilitates sleep. The brain is not passively receiving these signals. It is actively synchronizing with them, matching its own rhythm to the external one, slowing down its internal tempo in response to a world that has gone quiet. The brain that is now growing quieter as you listen is doing something it can only do in sleep. The sharp wave ripples are beginning to form in your hippocampus, replaying fragments of today, compressing them, sending them forward to the neocortex to be woven into the fabric of what you know. The sleep spindles are coming, bursts of thalamic activity that will punctuate the slow waves and carry information from the hippocampus to the cortex. The glymphatic channels are opening, the interstitial space is expanding, and the cerebrospinal fluid is beginning its quiet circuit through the brain, carrying away the molecular residue of wakefulness, the adenosine that has been accumulating since you woke this morning and driving your increasing sense of tiredness, the amyloid beta fragments that will, if allowed to accumulate night after night for years, slowly compromise the brain's function and the other metabolic byproducts of a day spent thinking, feeling, remembering, and deciding. The amygdala is quieting. The prefrontal cortex, which has been managing your decisions and regulating your emotions throughout the day, is relaxing its tonic control. The locus coeruleus, the brainstem nucleus that releases norepinephrine and maintains alertness throughout the day, is slowing its firing. The ventrolateral preoptic nucleus and the hypothalamus, which drives sleep by inhibiting the arousal systems of the brain, is becoming more active, releasing GABA onto the wake-promoting neurons of the brainstem. tilting the system from wakefulness toward sleep. It is not that the brain is shutting down, it is that the balance between two competing systems, the arousal system and the sleep system, is shifting toward the one that has been held back all day, the one that has been waiting. The brain is a universe of its own, and like the universe, it is mostly unknown. It contains more synapses than there are stars in the Milky Way. It processes more information than any device humans have built. It generates the experience of being here, of listening, of understanding, of existing. And right now, it is preparing to do the thing it does best, the thing evolution has refined over hundreds of millions of years: rest, consolidate, clean, and prepare. Good night. The slow waves are beginning, one by one, and the brain is going home, and you are safe inside it.